Hello everyone, my name is Bahgat Fayed. I am a postdoc fellow at National Research Center. And within 6 minutes, I will present only one of the projects that was carried out during my Fulbright fellowship in LA Biomed UCLA. I was working in two different projects. The first one is nanoparticles versus Candida Biofilm, new approach for combating antimicrobial resistance. And the other one is inducing gene knockout into multi-drug resistance Acinetobacter baumannii HUMC1. I will present only the first one, which is nanoparticles versus Candida biofilm. Let's start with the first keyword here, which is nanoparticles. Nanoparticles are any particles in the size range of 1 to 100 nanometers. We concern about this particle size range because the material as this size range has completely unique, different physical and chemical characters. If we look to the other keyword in the presentation here, which is Candida biofilm. Candida is an opportunistic microorganism that's capable to aggregate under certain conditions and coat itself with extracellular matrix, or what we call biofilm. Candida biofilm right now is considered one of the most microbial resistance form right now. It can tolerate the very high concentration of the antifungal drug up to 1,000-fold. Candida requires solid surface so that it can aggregate and grow and form the biofilm. That's why it's usually observed on the surface of the medical equipment, such as the casters, the implants, the heart valve. So the main resistance mechanism of the Candida biofilm is related to the complex nature of the extracellular matrix, which prevents the antifungal drug from penetration and reach the cells inside the biofilm. That's why the treatment strategy is based on two main mechanisms. The first one is to screen, look for new antifungal drugs, compounds that can disrupt this complex nature of the extracellular matrix and manage to reach the cells inside the biofilm and kill the cells. The other strategy is to look for drug delivery system where we can entrap the classical antifungal drugs inside the nanoparticles, and then the nanoparticles will be able to penetrate the Candida biofilm and release the drug inside the biofilm as the site of the action. That's why the project aim for my work was to fabricate different types of polymeric nanoparticles and see which nanoparticles will be able to penetrate the biofilm and release the drug inside the Candida biofilm. So as a first stage, I have fabricated different types of polymeric nanoparticles using the classical way of double emulsion techniques. And after that, I characterize the nanoparticles by scanning electron microscope to check the size. And this some image showing the nanoparticles which successfully managed to penetrate the biofilm. It has a size range of 100 nanometers. After that, we stain the nanoparticles with a fluorescence dye like the limousine red dye. Then we load the nanoparticles on the surface of the Candida biofilm, preformed on 96 well plate in lab. And we track the penetration of the nanoparticles inside the biofilm by confocal microscope. And this image showing the confocal microscope where one of the nanoparticles successfully managed to penetrate the Candida biofilm. So the nanoparticles stained in red successfully managed to penetrate the Candida biofilm within only 2 hours at different stage. The nanoparticles which successfully managed to penetrate the Candida biofilm was formed from a polymer called PLGA, PEG, which is the abbreviation for polylactic glycolic acid linked with polyethylene glycol. The PLGA is one of the polymers which is formed from different companies, especially Sigma, and it is approved by the FDA to be used in food and in drugs. But when we fabricate the nanoparticles only in PLGA, the nanoparticles fail to penetrate the Candida biofilm. But after we add the polyethylene glycol, the particles manage to penetrate the Candida biofilm. We think the reason is PLGA nanoparticles has a charge, and in the same time, Candida biofilm surface is charged. So we think there was like attraction or repelling because of the charge of the particles and the charge of the biofilm. But when we apply the polyethylene glycol, the particles become neutral. So we think the particles, when became neutral, it managed to penetrate the biofilm because it escaped from the charged surface of the Candida and go through the water channels located on the surface of the biofilm. I'm happy that after I returned back, we're still in cooperation with the American side, as we have applied for a co-fund from National Academy of Science in US and the STDF, and we are working on loading now drugs inside these nanoparticles. We have chosen drugs, alixidine and voriconazoles, and we have good entrapment for both drugs now in nanoparticles, and now we are checking the efficacy of the nanoparticles on completely removing the biofilm in vitro and in vivo, because the US site have modified mice castor model where we can evaluate the efficacy of these nanoparticles in vivo in mice. We are trying to see both effects, the treatment and the prophylaxis. So we will try to cause the nanoparticles on the mice and see if the nanoparticles will be able to protect the castor from the infection by Candida, and we will try also the treatment by loading the nanoparticles on castor already has the Candida biofilm on mice, and we will see its ability to completely remove the biofilm. Thank you.